And I would like to thank you for arranging this day and introducing all of us to each other. It's been great till now, so thank you. Okay,
my pleasure, my pleasure.
We can go to the next slide.
Sure.
So I will give you a brief overview about the idea behind Algae4IBD and the partners, Algae4IBD objective, the concept and approach, and expected outcome and impact. So we can go to the next slide. Next one. Yes. So Algae4IBD was established. The main goal is to combat or try at least to take algae and bring it into the world of inflammation, pain in general, but tackle the challenges of inflammatory bowel disease, specifically. And we would like to do that by developing either taking these algae-based ingredients, compounds, as a functional food or medication against pain, inflammation, and IBD. And why is that? I'm an immunologist and a biochemist for the last 25 years. And in those years, what I'm trying to figure out is how to establish novel approaches to tackle especially inflammatory processes, as inflammation is unfortunately the base of almost all disease you can even imagine. Either it's initiating that or it's making it worse. Autoimmune disease, cancer, and so on. Of course, chronic inflammation, it's a focus in my lab. So this consortium, this idea to establish the consortium and to answer the call came out because of the results we got from our lab, and together with other partners that I will mention in the next slide. Also, we have very interesting and useful results to bring it to IBD patients. So the consortium was built with 21 partners across Europe from 11 European countries, comprising 12 SMEs, industrial companies, mainly focusing on the biotechnology of algae and cultivating them or producing them, 7 academic partners and 2 IBD centers. Especially this multidisciplinary approach is to attack both the algae biotechnology and its challenges, cultivation, extraction, bioactivities, assessments, and exploring the compound discovery that relates to that and to synthesize it if we are lucky, preclinical assessment, novel food and food application by a food institute and food specialist. And the idea behind it is to take the whole pipeline that I will show you in a minute from A to Z, bringing it through the all research pipeline and preclinical assessment that is needed in order to get to achieve our goals. Next slide, please. Kostas, can we go to the next slide?
Yes, can you see the objectives? Change the slide.
Yes. So I will go.
Yeah, I'm sorry, there's a small delay.
No, thanks. Thank you. So I will go briefly of our objective and how we are trying at least to apply it in our work plan. So first of all, we would like to create a diverse ecosystem based on algae strain bank that will be specifically to Algae4IBD consortium with freshwater, marine, micro and macroalgae and cyanobacteria. Then for the small cultivation and extraction. And the main goal, as I said, the biodiversity of both micro and macroalgae is to observe and identify potent bioactive compounds to combat IBD at the end. Identifying and analyzing the correlation between the algae biodiversity and their potential bioactivity against inflammation, pain, and IBD, using high-throughput screening methods, which I will soon mention it in our work plan and how we are going to do that. Evaluate the effect of bioactive compounds derived from the algae for intestinal microbiome, and understand also antimicrobial properties for using it as a alternatively preservative for the food that we will produce at the end, to evaluate algae bioactivity in preclinical studies, like I said, as a preventive approach and as a treatment approach, and I will explain that in a minute. Scaling up this algae cultivated that we will choose by the end for the product prototype, and to apply the biorefinery approach, which will be very important at the end in the chosen ones that we will think that will be successful later on into the industry, and to assess the techno-economic visibility and life cycle assessment for the most promising products and processes. This is for the research objective. We have the business and social objective, like mentioned in the other project, building the business strategy and the IP that needed for that to protect the knowledge, to ensure international outreach, especially with critical target groups that are looking forward to see novel approaches, that those are the IBD patient, but not only, also researchers in the field and other policymakers for algae and how we can use it, and of course ensure public awareness, not only for IBD patient, but the algae usage and its advantage. Next slide, thanks. Next, yes. So as I said, our concept and approach in this project is to utilize the algae, both micro and macro, and to use it as antibiotics, prebiotics, pain relief, immune shield or immune promotion, and anti-inflammatory properties. Next. And the approach is it's a kind of holistic approach, because we would like to think in both ways. While we are exploring the pipeline, the research pipeline from in vitro and through in vivo, and at the end by the products, we are aiming in two kind of avenues. One from nature to plate, meaning we are going to produce 4 different categories of smart food, functional food, like bread, yogurt, gummy bears for kids, and another one, I don't remember now. And the other one is from nature to bedside, that it's more like more related to the drug discovery, if we will be lucky, of course, with the active compound that we can easily synthesize if we will be able to identify a compound that could be relevant to the pharma later on. Next, please. Okay, so I will quickly go over the steps, and then the next slide will be the one that will show you how it incorporates for the different work packages that are included in this project. So the first step is, of course, to identify and to establish a bank for Algae4IBD, which was contributed with all partners that are involved and are experts in the field from Europe and in Israel. So we had established more than 1,000 algae bank, especially for this consortium. And then we have a selective part that we identifying 150 algae that will be introduced to this project with all the steps needed in the pipeline, from more studied and more known algae, and the one that are understudied one, to open the field and to help us to discover novel compounds. Then we have the extraction methodology that we need, and then at the end we will use also biorefinery one. We need to test the compounds in different settings, diverse settings that could combat and the IBD challenges from different angles, and I will in the next slide I will explain more about that. Preclinical trials, we have in vivo mice models and ex vivo that comes from the IBD centers that contain patients from the age of 2 till the age of 60, compound characterization by HPLC, LCMSMS, and so on. Of course, NMR to help us to identify 1 or 2 compounds that could be synthesized later on. And the compound production or the fraction production or the extraction that is needed, it depends what the result will bring, whether we have synergistic effect that we need the certain fraction or certain extraction, or hopefully to get to a single compound or 2 compounds that can go to the farm. Next, please. Costas, maybe I'm in delay, but I need the next slide. Thanks. Okay, so how our work package is, the work plan is established is by, it's 10 actually work packages. The 10th was the ethical one that was included by the EU Commission after we were founded. So the first work package will be all about how to establish the algae bank, identifying the 150 that we want to address to. And we have like here we have also 2 kinds of approaches. We will already have like 20 what we call pre-indicated algae that are less risky because the results already came from my lab and at least 2 other labs that are participating in this consortium, already have the good indication, valid indication that we have a promising results as an anti-inflammatory or as a prebiotic that can go further on in this pipeline with less risk and more successful approach, at least that we will gain either compounds or functional food at the end. But we have also 130 algae that are under studies, and the beauty about that they are coming from different kind of conditions, either from volcano or hot waters or lakes around Europe and Israel that have the potential, according to what we think, according to how we process the data about them, the initial data at least, that could be a promising, at least knowledge-wise at the end, if it will be how to cultivate them or other challenges that we will discuss later, this would be one of the aspects. So we have a less risky one and a bit more risky one, but it will open the field at least. And then under these work packages, it will be all about how to cultivate them. For the pre-indicated one, we have a good protocols for that, but for the understudied one, it will be something that we need to cope with and to see how well they will be cultivated or not. We are going to process that, extract them, and later on be feedback with all the results that will combine with all the research work packages, and to build also strategies to optimize the production of the algae for other aspects as well. So next, it will go through the work package 2, meaning we have the bioactivity screening to help us to assess at the checkpoint which one will go next to the preclinical studies. So we are using one of the partners located in Prague. It's a specialist in high throughput screening for anti-inflammatory properties. Then this first screening for all these extracts that are done for each of the algae are going through this with the validation of specific anti-inflammatory properties in our lab and in other lab in Portugal. Then the testing of supportive beneficial bioactivities. It's not only needed to show the anti-inflammatory properties, but other one like antioxidants and so on. We have the techno-functional test because at the end, like I said, one of the approach will be a smart food. And we have also a digestibility assay to be sure that it will be tested before and after, kept the activity needed. And then parallel to that, we are doing compound identification, tentative identification, that at the next step, the one that will be chosen, we can also know what was the compound profile or composition. Then we have the work packet 3 will be all about focusing about the microbiota and antimicrobial action. So we are did over there already the screening for what kind of methodology and what kind of assay we want to test that's relevant to IBD. And then we are having the in vitro studies. We have the microbiome studies in mice model and microbiota studies also in zebrafish. Work package 4 will get the one that already been assessed in work package 2 and 3, and then will be tested. The most promising one will go through there, and this will be all about mice model, preclinical model, tackling colitis and Crohn that are both under the IBD umbrella, and then we will see what kind of pharmacokinetics, toxicity, efficacy that we are getting. And we will have additional aspect. It's ex vivo based 3D and 2D assays that will be derived from IBD patients from 2 IBD centers, one in Milan, one in Naples, having a range of patients from 2-3 years old till 60. And then we can do 2 things we can get for them. First, to see the efficacy, and then to see like if we can do it like personalized through age breakage or status and so on. Work package 5, it's a synthesis and algae improvement. That meaning that the most promising one are getting here, and we are doing 2 kinds of things. One, while we will identify a compound, we will start to synthesize that to have something at the end to bring it to the pharma later on. The other one is the algae that will be chosen, that chose the most effective one, at least the microalgae. I'm addressing the microalgae one. We can improve it in a non-GMO strain that is already done in CCMR in Portugal. They have the knowledge and the expertise, and then we can improve this kind of fraction or composition to make the algae, to the industry algae, something that they will have an improvement, a better algae to be used later on for the food industry, for example. Work package 6 is all about cultivation and the product development that I just said about the food. We have the upscale chosen algae that will be chosen. We hopefully we have up to 4, 2 from the microalgae and 2 from macroalgae. Those will be scaled up for the product development. We will have product formulation for an encapsulation, also for a proof of concept for a food supplement, and the food product that I already mentioned will be prototyped and developed by Work Package 6. Work Package 7 will explore all the techno economy analysis, exploitation, market and business development, and regulatory compliance that we need. Work Package 8 is all about dissemination and communication, that this is led by ESKI, doing all the engagement needed to be visible, to connect with the right stakeholders, especially with the IBD patient, but not only. And Work Package 9 is the management, and Work Package 10 is the ethics that we were added after we were founded. Next, please. So, expected results. So, 2 main goals that we would like to reach, or results that we would aim to reach. First of all, is gaining novel knowledge and for understudied algae, and novel bioactivities for commercialized one. And I'm saying that because when there are some studies about the ones that are out there already known, like spirulina or like ulva and so on, but even in my lab, a year ago, we have published that the spirulina that are undergoing certain biotechnology improvement have the anti-inflammatory properties, while the spirulina that cultivate normally and in the market for its protein or vitamins do not have these anti-inflammatory properties. So by doing all this research pipeline through all these work packages, we will validate and confirm the right properties needed for using that as a anti-inflammation, pain, and of course, with the usage of a certain purpose like IBD. Next one. Expected results, which we are crossing fingers that those are the results that we will really gain. The functional food, like I said, we had different categories aiming for kids and the general population for that could not only bring relief to IBD patients, but also to be like an immune shield, like in everyday life. If we will consume smart bread, it's something that will assist us maybe to prevent inflammatory occurrence, especially in the light, by the way, of COVID-19. All of you are already the 2.5 years already known the mentioned cytokine storm and so on. Food supplement, of course, that goes without saying, and the pharmaceutical one that will be based on a compound that we will discover that will be helpful for those purposes. Next, please. And the impact is really diverse, especially when we are focusing on IBD. IBD is a chronic inflammation disease that only raises and increases in the last 20 years. Unfortunately, the age which was used to be around starting at 15, that already got younger, is getting even younger. And now there is the age that we are experiencing that with the patient getting to the hospitals, it's already 2 and 3. And this is raising a lot of concerns, but it's not only in concerns in how the well-being of the patient. It's a burden on society. It's a burden of the hospitals, of the healthcare cost. It's also for the economic revenue, because those kids are skipping school's door. Those adults are skipping many, many days of working days. I'm not talking about the psychology and their social difficulties they are experiencing, but this is for that part, but also for the biotech, the algae biotech industry to reach to novel avenues and to get maybe commercialize this novel algae, or to open the field for them, the market for novel practice. This would be a huge impact, I think. Next, please. I think I'm done. Yes. Thank you for listening, and I will be happy to answer any question. Thanks.